everyone. In this particular video recording, I will be discussing the names of the scientists and their significant contributions in the discovery of DNA. So, who really discovered the DNA? Now, our basic understanding of the discovery of DNA was mainly founded or was established because of Francis Crick and James Watson. And so, when we are asked, in most cases, about who really discovered the DNA, what comes to our mind are the names of these two scientists, James Watson and Francis Crick. However, I'd like to highlight in this discussion that there are many people that have contributed actually to our understanding or to the discovery of DNA. And one of that is Rosalind Franklin. Okay? Now, dili lang po si Rosalind Franklin class ang ma-associate nato nga nakatabang para ma-discover ang DNA. Because, in reality, daghan kaayo sila ng mga scientists na nagtinabangay, each with their own timeline, para ma-discover ang DNA. One of them, we have Johann Friedrich Miescher. We also have Phoebus Levine. We also have Erwin Chargaff, Oswald Avery, Colleen MacLeod, Maclean McCarthy, and Fred Griffith, Alfred Hershey, Martha Chase, William Asbury, and Morris Wilkins, Rosalind Franklin, James Watson, and Francis Crick. So now, let us learn what are their significant contributions, like each of them. Husay gyud ang ilang natabang para mas mahanay and para mas strong, para mas mapadaghan pag gyud ang information about DNA. And para ma-discover, matagaan pod og kanang importance ang discover sa DNA. Let us start first with the significant contribution of Johann Friedrich Miescher in the discovery of DNA. Now, Miescher class is a physician and actually fascinating kayo ang iyahang passion or iyang interest because he really likes to study the chemistry of pus. Now, what is a pus? Ang pus class kay mo ni siya ang or siya nuka kanang fluid ba, this thick yellowish or sometimes greenish fluid that is produced as a result of infection. Iba kabante mo ana nay butoy sa ato ang mga panit baron or parts sa ato ang lawas, and then usahay ato na siyang pisaton, and then ay mo gawas na yellow nga liquid. So mo na siya ang pus. And usually ang pus ma-develop na siya sa atong lawas because of our response, no? So ang reason nga nung na atay mga pus, it's a response sa atong lawas against mga bacterial invasion, no? And pus is also a combination of white blood cells or mga leukocytes, even mga dead cells or mga living or dead na mga bacteria na nasa area. Okay, so that's a sign of infection. That's a response of our body against mga microbial invasion. So, yun, that's the interest of Miescher. Ganahan kayo siya nga mag-study about pus. And a reason po nga nung ganahan siya mag-study about pus is because ang pus daw is daghan kaayong mga white blood cells nga dagko og nuclei or katong nucleus ba. And so, since mo man to ang iyang field of interest, he was able to isolate the nuclei of white blood cells that are found in pus. And then he called that substance nga iyahang or nuclei nga iyahang na-isolate gikan sa pus as nuclein. So siya gyud ang una nga naghatag og pangalan sa DNA, in which he called that one as nuclein. Nga naa daw substance na nuclear material nga naa sa white blood cells. And then, he also proposed, or he also provide further information about this nuclein, and he said that this nuclear material is slightly acidic and is high in phosphorus. So, aside nga naghatag siya og pangalan sa substance nga iyang na-isolate from the white blood cells, iya pa diyang gi-describe di ay kung unsa ang characteristic atong nga substance or atong nuclein, in which he proposed or he said that nuclein is slightly acidic and high in phosphorus, which is very true. Because if you're going to study DNA, we will come across to an information that would describe DNA as acidic, and they have phosphate group, which is part in their monomer, in their building block ng mga nucleotides. So that's the contribution of Johann Friedrich Miescher. Now let us go to Albrecht Kossel. Unsa pud ka ang iyang contribution? Now Kossel class carried out further work on the chemistry of DNA, and he was able to determine that DNA DI contains 4 nitrogenous bases. We have adenine, cytosine, guanine, and thymine. So that's the contribution of Kossel. Now let us move on to the contribution naman of Phoebus Aaron Levine. Now, si Levine class, he discovered that DNA is consist of nucleotides. So he described nucleotide to be made up of phosphate, sugar, and a base, or a nitrogenous base. So that was the idea of, or that was the discovery of Levine. He suggested, or he proposed that, or he discovered that DNA consists of nucleotides, and that each nucleotide contains a sugar, a phosphate, and a nitrogenous base. Then aside from that, aside nga niingon siya nga ang DNA is made up of nucleotides, si Levine class also proposed the concept of tetranucleotide hypothesis. So what do we mean by tetranucleotide hypothesis? So ingani ang representation sa iyang hypothesis, in which actually this is an incorrect. So incorrect ni siya nga hypothesis class ha. Pero ato lang po tagaan og kanang highlight or kanang appreciation, since during their time wala baya gyud sila mga high-end technology to be used para maka work sila at the molecular level sa DNA. But then they have this idea nga so they're very creative and intelligent in their times. So Phoebus Aaron Levine, he proposed the tetranucleotide hypothesis, in which, according to this hypothesis, the DNA is consist of 4 nucleotide units, and each containing all 4 bases. So pasabutan na, di ba, according to him, ang nucleotide kay made up of sugar, made up of base, a nitrogenous base, and phosphate. So ang tetranucleotide pasabutan na ang DNA daw upat lang ka nucleotide ang nag-compose ana niya, and then in each of the nucleotide naanay upat ka or naatong upat ka nitrogenous bases, the adenine, cytosine, guanine, and thymine. So this is actually incorrect because we have learned now that in every nucleotide, isa lang gyud pod ka base ang naa diha. Okay? So that was the contribution of Levine. Now let us move on to the contribution of Erwin Chargaff and his colleagues. Now, Erwin Chargaff and his colleagues measured the amounts of 4 bases in DNA from a variety of organisms. And they found that DNA from different organisms varies greatly in base composition. So, sila ang nagpamatuod nga ang mga bases nga naa sa matag organisms kay managlahi. And so because of their discovery, they disproved the tetranucleotide hypothesis. Because according man good sa tetranucleotide hypothesis, fixed na ang DNA is made up of 4 nucleotide which also has 4 nitrogenous bases. And sila Chargaff, sila ang napamatuod nga matag klase sa mga organisms, lahi-lahi pod ang number sa ilahang base, ilang nitrogenous base composition. And in particular, Erwin Chargaff and his colleagues discovered that within each species, there is some regularity in the ratios of the bases, which means that the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine, which is why we have the Chargaff rule, which denotes that the number of a particular nitrogenous base is always equal to the number of its complementary base. For example, if adenine, we have 36 adenine, then that would mean more or less na po 36 ka thymine. So that's the Chargaff rule nga. Equal ang ilang ratio regardless of their characteristic. Basta always equal ang ratio sa mga numbers sa atong mga nitrogenous bases, especially that they are complementary bases. They will always pair and they will always complement with each other. Okay. So for example, in the case of E. coli, no? As you can see here, this is the percent in moles of nitrogenous constituents per 100 grams atoms of phosphate in hydrolysate. So in E. coli, for example, class, the number of adenine is 26 and then the number of thymine is 23.9. So, same sila. And not same, okay, of course 26 ang isa, but more or less duol sila ba? Okay? Then, ang guanine, 24.9, cytosine is 25. Ang yeast, mapo ni ang number sa adenine. Si urchin, mapo ni siya, thymine. So, as you can see here, their ratio is more or less 1, which only means na the number of the bases is almost equal, or if dili man equal, duol lang po sila nga pagka-equal with the number of their corresponding complementary bases or complementary base. For example, ang adenine or thymine, duol lang sila sa number po with the adenine base. Or guanine pa ron, di same pa po siya, or almost same lang po sila with the number of cytosine. So that's the discovery of Erwin Chargaff and his colleagues. Okay. They were the ones who suggested that there is a regularity in the ratios of the bases, and the amount of adenine is always equal to the amount of thymine, and the amount of guanine is always equal to the amount of cytosine. Now let us proceed with the discovery of Fred Griffith. So actually we have a very nice experiment of Fred Griffith. We call that one as the Griffith's experiment, in which in his experiment he explored the virulent and the avirulent type of Streptococcus pneumoniae. So he was the one actually who used the Streptococcus pneumoniae because he was a physician and he was able to isolate different strains of Streptococcus pneumoniae. And so we will have a separate discussion of his work, and so let's focus lang muna about his significant contribution. So Fred Griffith conducted an experiment with Streptococcus pneumoniae bacteria, and he was the one to first reveal the transforming principle, which led to the discovery that DNA acts as a carrier of genetic information. So si Fred Griffith, siya ang nag-formulate ng idea nga transforming principle, in which from there maka-conclude ta nga ang DNA is the carrier of genetic information. And then we also have Oswald Avery, Colin MacLeod, and Maclean McCarthy. So, because of the work of Fred Griffith, daghan kayo ng mga scientists na naa sa scientific community ang skeptical about DNA. Because most of them would believe that the carrier of genetic material is the protein and not the DNA. And so, these three scientists, Oswald Avery, Colin MacLeod, and Maclean McCarthy, conducted further experiments to really prove if was it really the DNA or was it a protein. And they succeeded in, so it's more of like, ang ilang experiment class kay more siya sequel or more siya further tawag aning experiment siya ba nga mag-further prove about the truthfulness of DNA serving as the carrier of genetic material. Pero maingon ako nga ang ilang research kamo siya sequel sa research for ni Fred Griffith. Ang ilahang MacLeod kay murag part 2 sila. Kay very interested mangod sila kay during that time wala si Griffith mangod ang pinakauna nga nag-ingon nga naay something, naay transforming principle or naay reason nga nung na-transform ang katong virulent strain into virulent, especially if katong pag-combine nila sa heat-killed nga virulent strain and then katong avirulent R strain. So, basta kato siya, madiscuss na nato later on. So, na-intriga sila ato ba? And so, nagbuhat pa sila ilang kagaling experiment. And their research, their contribution was that they succeeded in isolating and partially purifying the transforming substance. And they showed that it had a chemical composition that is closely matching that of the DNA and quite different from that of proteins. Kaya lagi, before, most of the scientists in the scientific community believe nga ang genetic material are the proteins and not the DNA. So they provided compelling evidence that the transforming principle or the transforming substance nga nakuha po nila dito sa research or dito sa experiment ni Fred Griffith was therefore genetic information that resides in the DNA. So sila ang nihatag og further evidence or nihatag sila og kanang another evidence ba para mo prove nga ang DNA gyud siya ang carrier of genetic information or ang genetic information makit-an dito sa DNA. So that's the discovery or that's the significant contribution of Avery, MacLeod, and McCarthy. Now we also have Alfred Hershey and Martha Chase. So these two scientists actually, they demonstrate that DNA is the genetic material in the T2 bacteriophage. So kani sila na scientist, kani silang duha, sila ang nahaghatag og another compelling evidence about the truthfulness of this idea nga ang genetic material is makit-an siya sa DNA. Okay. And the research was on the particular special type of virus, the T2 bacteriophage, which during their time, they used the T2 bacteriophage in the experiments manggod po, which is we have the Hershey and Chase experiments. Nga ito po siyang i-discuss separately. Pero basically, ang ilang gibuhat dito plus kay ilang gi-infect ang E. coli with a T2 bacteriophage kaya para matrace yun nila kung kinsa gyud ang ka-carry sa genetic material. Is it the DNA or the protein? So that's the contribution of Hershey and Chase. Now we also have William Astbury. And William Astbury, he began studying the three-dimensional structure of DNA by using a technique that is called as X-ray diffraction, in which the rays beamed at a molecule are reflected in specific patterns that reveal aspects of structure of the molecule. So siya ang pinakauna nga scientist nga nigamit og X-ray diffraction para makastudy sa three-dimensional structure of DNA. However, it's just that his diffraction images did not provide enough resolution to reveal the structure of DNA. Now, we also have Maurice Wilkins. So, kani si Maurice Wilkins class, kauban ni siya nila James Watson and Francis Crick nga nahatagan og Nobel Prize for their discovery on the three-dimensional structure of DNA. And Maurice Wilkins also used X-ray diffraction to study DNA. Now later on, you will know what is the connection of this Maurice Wilkins with Rosalind Franklin and sa iyang nabuhat before nga makaingon ta nga dapat wala unta ito niya gibuhat because that's dishonesty or stealing of intellectual property. Okay. And then we also have James Watson and Francis Crick. So sila gyud ang nihatag class or ni-provide and ni-demonstrate sa three-dimensional double helical model for the structure of DNA. So, sila gid pod ang dili pod nato maingon nga dili nato sa taga-credit kay sila man gid ang napamutuod nga ang DNA kay ang three-dimensional model sa DNA kay double helix siya. And it's just that nalang pug ko'y gamay nga kanang tawag aning dislike sa pamaagi as to how they give credit sa mga tao nga nakatabang sa ila because they claim man gud nga sila lang ang ilaha lang own na mga pagpaningkamot ang ilahang ang main reason ba nga naka-formulate sila aning three-dimensional model sa double helix when in fact naa sila gigamit nga information from Rosalind Franklin. Okay. So, basta mo to siya ang contribution ni James Watson and Francis Crick. Now, let us go to another scientist. No? She's actually the first woman to more maingon na to siya ba nga among all na mga scientists nga nakatabang para ma-discover ang DNA. si Rosalind Franklin ang yun ang nalahi because first, of course, she's a woman and she really is very intelligent. Like, lahi yun ang iyang caliber class. Kaya nga dyan nga polite yun siya. So, si Rosalind Franklin, she worked in Wilkins Laboratory, which is why some accounts would claim nga usahay dili sila magkasundo ni Maurice Wilkins kay isa ra sila og kanang laboratory or lab nga gigamit. And Rosalind Franklin class also use X-ray diffraction. So, he works in Wilkins Laboratory and she obtained strikingly better images of the DNA molecule, which is we call this image here, as the photo 51. So, napo siya kauban na estudyante actually nga nagkuha or nga kanang nitabang sa iya ba para makakuha ani nga photo 51. Pero among all na mga scientists nga nigamit og X-ray diffraction, si Rosalind Franklin lang gyud ang nakakuha og nindot kaayo nga image sa DNA molecule. And the reason for that is because Rosalind Franklin is an excellent X-ray crystallographer. So, expert na gyud siya kung saan paggamit, and then kabalo siya ba sa iyang ginabuhat, saan pag-arrange ang ilahang mga specimen siguro para maka-come up sila with a better image. And her images actually were critical in the eventual elucidation of the structure of DNA by Watson and Crick. Which is why kaganina I have made mention nga in a way mura kog wa ganahi sa pamaagi as to how Francis Crick and James Watson give credit to themselves in the discovery of the double helix, the three-dimensional structure of the double helical model. It's because they claim manggod nga sila rang tulo, si Maurice Wilkins, si Francis Crick, and si James Watson ang naka-discover using their own unique ideas. When in fact, nigamit sila og image kaning photo 51 nga nakuha ni Rosalind Franklin. Okay, so actually class, complex kay ang story ani. And for you to be able to appreciate the contribution of Rosalind Franklin, which is the unsung hero of DNA, so I'd recommend you to watch this video, which is a TED Ed ni siya, and was published in the year 2017. And ma-research tapos din ninyo sa YouTube. So, I'd recommend you to download kanang watch this video para ma-appreciate po ninyo ang contribution ni Rosalind Franklin sa discovery of the double helix. Okay. So, Watson and Crick, they investigated the structure of DNA not by collecting new data, but by using all available information about the chemistry of DNA to construct molecular models. And so by using the excellent X-ray diffraction images taken by Rosalind Franklin and by applying the laws of structural chemistry, they were able to limit the number of possible structures that DNA could assume. And so they tested various structures by building models made of wire and metal plates, and with their models, they were able to see whether a structure was compatible with the chemical principles and with the X-ray images coming from Rosalind Franklin. So actually class, kaning X-ray diffraction images or katong photo 51 nga nakuha nila kay Rosalind Franklin, wala kahibaw si Rosalind Franklin ato nga nakita dahito nila James Watson and Francis Crick. And ang reason po nga nung naabot kila Watson and Crick ang photo 51 is because gikuha to siya or gi-steal ni Maurice Wilkins kay, di ba? Same ba sila og lab no? So maybe na-invade siya sa kanang space or sa laboratory po ni Franklin sa area since the same naman silang building or the same naman silang lab sina siya gikuha yun or iyang gikawat no ang photo 51 tapos iyang gipakita kila Crick and Watson. So yun ang work nilang Watson and Crick. Pero I mean my point of view, class, is not to hate Watson and Crick but to look at it in a bigger perspective pa nga the scientific community before is male dominated, which is why if you're a woman in that era, maglisod gyud ka og patunay sa imo ka og galingon nga you are capable and that you are intelligent or you have the same capacity with your male colleagues. Kay lagi male dominated arena ang scientific community before. So luoy lang po kay story si Rosalind Franklin kay it's too late. Namatay na siya before nahatagan og kanang appreciation ng iyang contribution sa DNA. So I'd recommend you to watch that video nga akong ipakita kaganina para mahibawan gyud ninyo ang iyang or ang contribution ni Rosalind Franklin. Okay. And then we also have Hans Frankel, Conrad, and Bea Singer. So these two scientists also demonstrated that for few viruses, the RNA is used as their genetic material and not DNA. So before, gabi kayo sila og kana interest sa DNA. And then here comes sila Hans Frankel, Conrad, and Bea Singer, in which ilang gipatunay or naghatag pud sila og kanang another evidence nga dili lang all the time ang genetic material ka makita sa DNA. Because in the case of virus, they use RNA as their genetic material. So, ang reason po gano sila Conrad and Singer is naka-formulate aning idea is because they work manggod with tobacco mosaic virus or TMV. And they found that this virus possesses a single molecule of RNA that is surrounded by a helically arranged cylinder of protein molecules. So yun, sila nagpatunay nga for some dili man good organism ang virus, but for some virus or for most virus or for few viruses, they use RNA as their genetic material. Alright, so here is the timeline about the people who have contributed to our understanding of the structure of DNA, starting from Brown, which is, diba, siya man na nakadiscover sa nucleus sa cell up until nisulod na to sila Miescher, na sila Levine, sila Griffith, sila Cossell, sila Avery, daghan na kayo up until in 1956 sila Conrad and Singer, in which dito na natagaan pong highlight ang RNA as the genetic material for some viruses. Alright, so that ends our discussion about those scientists or those people who have contributed to our understanding of the structure of DNA. And I do hope that you appreciate this scientist, even though daghan kayo sila. And yun. So that ends our discussion for today, and see you next time. Goodbye, everyone.